My name is Jordi Merino. I'm an associate professor and group leader here at CBMR. Welcome to everyone. And I'm today delighted to introduce the next speaker, Eran Segal. Eran is a professor at the Department of Computer Science and Applied Mathematics at the Weizmann Institute of Science in Israel. His research focuses on how genetics, gut microbiome, and nutrition affects health and diseases, with a special focus on developing bioinformatic tools to better understand the relationship between health and disease. His aim is to develop personalized medicine strategies based on large-scale and deep phenotype cores. Eran has been highly successful in publishing at high top journals. He published more than 200 manuscripts and has received several awards and honors, including the Overton Prize and the Michael Bruno Award for his contributions to the field of bioinformatics. And with all of this, Eran, I'm delighted to have you here.
Thank you. Yeah. Thank you, Jordi, and thanks the organizers for the kind invitation to speak to you today and tell you about a large-scale population cohort that we've been collecting. We were motivated to do this by seeing other data collection efforts, starting from the Human Genome Project, which, if you look from the year 2000 until now, has been really a transformative project, taking us from knowing just a handful of genetic variants involved in some diseases to where we are today after profiling and genotyping tens of millions of people, where we now know hundreds, if not thousands, of genetic variants for virtually every disease. But of course, the genome is just what we're born with and doesn't take into account environmental factors, exercise, nutrition, and so on. And for that reason, like we had the Human Genome Project, we decided to initiate the Human Phenotype Project to phenotype people very deeply, with the hope that this type of data would allow us to identify trajectories of disease and find novel disease biomarkers. And so to that end, what we did was several years ago, we started a clinic at the Weizmann Institute where before participants come in, they undergo, on the right-hand side, you can see they upload their electronic health records. They fill out hundreds of questionnaires about medical history, family history of disease, and so on. So we know all of their medical status. Then they come to the clinic. They undergo various anthropometric measurements, so various body measures, hip and waist circumference, hand grip strength, cardiovascular assessments like ECG, voice recordings, which correlates to disease, then various imaging modalities. We use DEXA to look at full body composition and bone density, high-resolution scans of the retina, from which we can see blood vessels. It's considered to be a window to the heart. We use ultrasound to look at fat in the liver and the carotids, also for cardiovascular health. Then various sensors that people go home with, continuous glucose monitors track glucose levels every 15 minutes, sleeping devices that track quality of sleep. I'll show that later in the talk. Smartwatches track physical activity. While people are connected to these CGM, continuous glucose monitors, they log all their dietary intake, which they're incentivized to do because they get a report of which meals spike and which do not spike their glucose levels. They track medication they take and physical activity, and we biobank samples. We have both live cells from everybody, PBMCs, and serum. And maybe the most distinctive feature is connecting all of this with the more advanced genomics and multi-omics testing, which includes whole genetic sequencing. So we also look at the human genome, but then also gut and vaginal, recently microbiome, metabolomics, which looks by mass spectrometry at thousands of different small molecules in the blood, proteomics from blood, RNA sequencing in bulk, also from the PBMCs, and a novel immune assay that we developed whereby we can synthesize hundreds of thousands of antigens and then see which of these are bound by a person's antibodies in the serum. So, if you will, this is a snapshot of the entire infectious history of a person. So all of this we did on over 12,000 people in Israel. It's a longitudinal cohort. People get profiled every 2 years. We can look at this data also and partition it by body systems. We're profiling 17 different systems of the body. On the right-hand side, you can see also some kind of very global map of how the features of different body systems relate to each other. This cohort has about 8,000 people who have one or more of these morbidities shown here, mostly cardiometabolic morbidities, and about 4,000 by exclusion for not having any of these are considered to be healthy. And it's very nice to be able to study on a standardized platform, everybody measured in the same way, and compare all of these diseases to each other. We also have about 30 what we call medical conditions, not diseases, but still interesting to study, like various vitamin deficiencies, allergies, and so on. Because we see the value of this data, which will be the main focus of the talk today, our discoveries, we're now in the process of trying to significantly expand this. With the next milestone, we hope to achieve is 100,000 people, both by doubling and tripling this cohort in Israel, but also expanding to other countries where we can explore more genetic, cultural, ethnic, and so on diversity. And in 2024, we're actually now building clinics in Abu Dhabi and Japan. We'll start to add these to the cohort. Now, of course, this is not the only large-scale population cohort. There are quite a few that are being carried out worldwide. The most impressive one, in my view, is the one in the UK called the UK Biobank. It started 17 years ago. 500,000 people profiled, also longitudinally, and the impact that this project had is really enormous. It really changed the way that we do biomedical research. 40,000 researchers from 100 countries are working on this data collectively. They published over 10,000 papers. You can see how it grew over the years, with close to 3,000 publications in PubMed just last year. In comparison, we obviously don't have a cohort that's as big, but we have several unique features, like it's going to be the first cohort to be international, as I mentioned, spanning multiple countries, and then all of these other measurements shown here, mostly the more advanced multi-omic measurements that we have that are not part of the UK Biobank, are unique features to this cohort, which is why I believe that this will be complementary to the UK Biobank. And like the UK Biobank, we are also giving access to the cohort free for academics for research. So in the remainder of the talk, I'll tell you about discoveries that we've been making from studying this cohort. And I'll go pretty briefly on each of these different projects, just so that I could cover and show you the breadth of what could be done with this type of data. And the first project I'll start with is something we actually started 10 years ago. It was even a precursor cohort to the large cohort, where we profiled 1,000 individuals with continuous glucose monitors and found that people have very different blood glucose response to food, as you can see by the 4 different curves here of the 2 hours after eating 4 slices of bread, very different between individuals, but lines of the same color correspond to the same person on different days, showing very consistent behavior within a person, different between people. And using clinical data and gut microbiome data, we showed that we could fairly accurately predict the actual personalized responses. Every dot is an individual meal, where the x-axis is the area under the glucose curve that's actually measured after eating that meal, and the y-axis is the predicted one according to the model. And then we could show that you could take such an algorithm and take individuals who are pre-diabetic. They have many spikes in their blood glucose levels over the course of a week. And with a diet that's equal in the amount of calories for every single meal, we could prescribe a diet that fully balances their blood sugar levels. After publishing this on a short-term intervention, we went and did a full-blown randomized clinical trial on 200 people with prediabetes, randomized them into either a standard of care Mediterranean diet or the algorithm diet. And you can see anecdotally, one participant before the intervention, many spikes in glucose levels at the top, and at the bottom, in the 1 month of CGM tracking after the intervention on the algorithm diet, virtually eliminating these spikes in glucose levels. And statistically looking at the primary outcomes in the center panel, hemoglobin A1C, which is the average glucose levels in the past 3 months. In the red curve, you can see an improvement on the Mediterranean diet after 6 months of intervention, but we get double the improvement on the algorithm diet. And on the right-hand side, we have multiple metabolic parameters that also improved in people following this diet, some of them improving more significantly on the algorithm diet compared to the Mediterranean diet. So after we published this, we were also interested in kind of going a little bit more deeper into gaining insights on the mechanism by which this dietary intervention actually had an effect on various metabolic parameters. And I'll show you a few analyses that we did that convinced us that at least part of the effects, part of the way in which the diet induced beneficial metabolic effects was mediated by the microbiome, namely by the changes that the diet had on the microbiome. And we showed this in two different ways. First, just by looking at all the intervention and what happened when you compare samples at baseline to samples after 6 months of intervention, we saw many changes at the center in gut bacterial species and at the right-hand side on different metabolites that we measure with mass spec through metabolomics. And then in previous work done on a different cohort, we showed actually that quite a lot of the metabolites, several hundreds of the metabolites, could be very well predicted by gut microbiome composition. So here's an example of 4 metabolites that on held out samples are predicted quite well from the microbiome composition of individuals. And so what we did was we took these models developed prior to this work and on a different cohort, and we applied them to the samples on the dietary intervention, both to baseline samples and samples taken after 6 months. And because there were changes in the microbiome, there were also changes in the predicted levels of certain metabolites according to the model. And we then compared them to the actual changes in the same metabolites as measured by metabolomics on the dietary intervention between baseline and post-intervention 6 months samples. And we saw a fairly good correspondence, meaning that we believe that some of the metabolite changes that we saw in people are in fact mediated by the changes that the diet induced in the microbiome, because we could predict these changes in the metabolites based on the changes in the gut microbiome. Another way in which we looked at this was by mediation analysis. And before doing the mediation analysis, we just looked at a direct relationship between how diet affects changes in the gut microbiome. And so for each gut bacterial species, we predicted its levels on the big cohort that we have of 12,000 people, predicted it based on dietary data that we had. And quite a lot of the gut bacteria we can actually predict from what people are eating, not with perfect accuracy, but with fairly definitely significant and fairly good Pearson correlations. And if you look at some of the best predicted species, you can see that just by diet alone, you could predict quite robustly what the levels of the bacterial species would be in individuals. And so then we could go and do what's called mediation analysis to try and get at causality, where in mediation analysis, you're trying to assess whether you have whether the relationship between the X, in this case diet, and the Y, in this case various metabolic improvements that we saw, are also mediated in part by some other factor, in this case the microbiome. So to do this analysis, you construct models that try to explain, in this case, the metabolic improvements from the diet. And you know that you have the microbiome, which also is affected by the diet, and you ask whether the microbiome also contributes to explaining these beneficial metabolic outputs beyond what you can explain with the diet. And we found quite a few of these cases. They're all summarized in this work. I won't go into the details, but several of the metabolic improvements, like, for example, the time spent above 140, and on the CGM, so how many spikes of glucose levels mediated by particular bacterial species and particular dietary components. So overall, by these two different analyses, we believe that at least some of the beneficial effects of the diet are actually mediated by the changes that the diet induces to the microbiome. Continuing on the thread of the microbiome, we also have efforts to try and develop direct therapeutics based on the microbiome. And for that, what we did and recently published is very analogous to what people do in human genetics, where the main method of analysis is called GWAS, Genome-Wide Association Studies, where you associate a genetic variation across individuals with a human trait of interest. So what we did was to develop and adapt that methodology to the microbiome, whereby we look at genetic variation, not on the human genome, but genetic variation on bacterial species at the single nucleotide level, and associate that with human traits. And so here's a result of that. This is a standard Manhattan plot that many of you probably saw from human genetics, except that instead of the x-axis now being human chromosomes, the x-axis is now bacterial species. Every dot corresponds to one particular position within a specific bacterial genome, and the y-axis corresponds to the strength of the association between genetic variation at that position and a human trait of interest. In this case, we're looking at BMI. And so overall, we found over 1,000 different of these SNPs. Many of them actually, they come in blocks. So after you remove SNPs that are correlated to each other, we're left with about 40 different independent positions in 27 different bacterial species that showed strong association. Interestingly, in 12 of these cases, if you look at the abundance of the species that encompasses, that contains the SNP, the abundance of that species is not correlated to BMI, but when you look at the genetic variation, it is. And I think even more interesting than just the strength of the association is actually the effect size that this could explain. So now I'm plotting exactly the same data. The y-axis is still the minus log p-value, the strength of the association, but now the x-axis is actually how many points of BMI are explained by each of these different SNPs, and you can see that there's quite a few SNPs that explain 1, sometimes even approaching 2 points of BMI that are explained by just a genetic variation at a single bacteria, one position. And this is even the size of these effects are stronger than some of the strongest effects that people have seen on the human genome. And the way I rationalize that is because I believe that the microbiome is an environmental factor, and we know that environmental factors have big effects on many different traits, definitely on BMI. And the reason we think it's an environmental factor, we did work several years ago where we showed that genetics explains very little of our microbiome composition. So on the cohort in Israel, when we do just a simple PCA plot on the left-hand side of the genetics, we can see a very clear partitioning of different populations in Israel coming from different locations in the world that are known to be genetically distinct. But when you look at the same microbiome composition, all of these populations that are genetically different, they're actually completely mixed in terms of the microbiome. And there were other analyses in this work and follow-up works also by others to suggest that the human genetics explains a very little portion. We estimate 2 to 4% of the microbiome composition, and the rest is environmental factors. You get your microbes from the environment. We also validated these findings on the BMI, the particular SNPs, on a Dutch cohort of a similar size. And we showed that the majority of these SNPs were also significant in the Dutch cohort, and almost all of them, with the exception of one, even if they were not significant, the direction of the association, whether it's a positive or negative association to BMI, was maintained also in the Dutch cohort. If we look at some of these SNPs individually, we can see, for example, here one of the most strongest associations. If you look at the center here, we have about 2,000 individuals that in this particular species had the major allele for this SNP. They have an average BMI of 25, and about 400 individual on the right that had the minor allele that on average have a BMI of almost 27, 1.9 points of BMI explained only when you partition people based on this SNP, so a very strong association. What's also nice about the associations that the resolution of SNPs is that you can look at the not just the bacteria, but the individual genes that are being affected. So here's an example of another SNP explaining 1.3 points of BMI, that if you look at this region, it contains various energy production genes, lipid metabolism genes that you might rationalize could be mechanistically involved in the way in which bacteria would have an effect on a trait like BMI. And another example in an inflammatory pathway also makes kind of sense that could be involved in a trait having an effect on a trait like BMI. Using exactly the same pipeline, I'll just briefly mention, we're also looking at association not to human traits, but to abundance of other bacteria. The reason is that this may allow us to find genetic variation in some bacteria that affect the presence of other bacteria, namely they could be some novel antibiotics. We also look at associations to the same bacteria that harbors the SNP that can allow us to identify genetic variation that help or induce or inhibit the growth of the same bacteria. And quite interestingly, just from a systems level perspective, we see that genetic variation within the same bacteria typically has positive associations to abundance of the same bacteria, namely evolutionarily, as you might expect, bacteria evolve their genomes such that the mutations support the growth of the organism, whereas if we look at associations with other bacteria, most of these are actually negative, supporting kind of the warfare that we know is going on between bacteria. And also the actual genes are very different. Genes in the same bacteria that are associated are mainly housekeeping genes having to do with growth, whereas genes involved in the association with other bacteria are mainly related to metabolism genes. Maybe in the interest of time, I'll skip this short piece and tell you now a little bit about the sleep data that I mentioned. So I think this is also quite unique data that we are collecting here. So there's a company in Israel who developed a device, FDA approved, for diagnosing sleep apnea. It has several sensors like actigraph, accelerometer, and another device on the finger that measures blood saturation. So overall, by tracking 3 nights of sleep from each participant, we get several different analog channels throughout the night from which we can extract multiple features, and something like this was never measured on a scale of thousands of people, definitely not when it's coupled with all the other parameters that we're measuring. So for example, we can see differences between males and females and how these measures of sleep apnea actually change with age. We see that they change with age, some known association. We also see that if you try to diagnose sleep apnea based on these, which is what the device is used for, there's a difference in the diagnosis people would get if they use it for 1 night or 3 nights, supporting multiple use, multiple nights in order to get robustness. And also we see different associations between all of these different sleep features and the multiple body systems that we have, such that we can use the sleep data to predict with some decent accuracy several measures about people having to do with sometimes their blood tests, sometimes food that they eat, dietary patterns, and so on, and also various associations with disease. And then the final project I want to tell you before a short last segment on AI models that we're building with this is looking at human aging. Because we have so many different types of data, there's an opportunity to see how this data changes in people with age. And so first we took just the metabolomic data, and we used the metabolomic data to predict the chronological age of people, which you can see here. The predictions are working fairly well. The y-axis is the predicted chronological age of a person using only the metabolomic data. The x-axis is the actual chronological age of a person. But you can also see that these predictions are far from perfect. People at the top have a predicted age, which according to the model is older. So when the model looks at their metabolomic data, it seems like to be more similar to somebody who's older. And conversely, people at the bottom according to the model, they have metabolomic data that resembles more somebody who's younger. And so we asked whether these errors in the model are actually just errors in the model, or whether they actually carry some meaning that people at the top maybe are actually aging faster. And indeed, when we compare people at the top to people at the bottom and look at other independent parameters that were not used as part of the model, we see that people at the top are in worse health condition. They have higher BMI on average, higher triglycerides, hemoglobin A1C, blood pressure, cholesterol, and liver enzymes. So, this model, which we call a biological clock, in this case a metabolomic biological clock, is also informing us of the clinical condition of people, and might allow us to identify biomarkers that are indicative of accelerated age, at least according to the metabolomics data. But we have many different types of data. So now looking at just the retina scans, the original image is this one. The other images are what a deep learning model algorithm applied to these images see in terms of blood vessels and various features having to do with their widths, their curvature, their fractal dimension, and so on that you can extract. And we can also look at how these parameters change with age, and some of them actually change with age. And more broadly, we can look at all different systems of the body that we measure in this cohort that I showed you before. And for each of these body systems, we can develop this model that tries to predict chronological age. All of these models that we developed had a significant predictive power for chronological age, some with better accuracy, some with worse accuracy, but they were all significant. Interestingly, we found that these clocks were by and large independent from each other, meaning that if in one individual, one body system is aging faster, it doesn't necessarily mean that another body system will age faster. This is a result that somewhat surprised us. The second key result that we found is that these clocks are clinically meaningful. Like I showed you in the case of the metabolomics clock, that people with a higher age were in worse clinical condition. Typically, people with a higher age in one body system were in worse clinical condition, but not just worse clinical condition, they were at higher risk typically for developing a disease related to the body system on which the clock was developed. So, for example, a clock developed for insulin resistance parameters, people who had a higher biological age were at higher risk for developing diabetes. And a final result that we saw was a marked difference between males and females, with males kind of aging pretty much linearly, but females having this abrupt change right between the age of 50 to 55, which we could link to menopause, because when we looked at women of the same chronological age, but comparing pre to post-menopausal women, we saw several biological clocks that were higher in postmenopausal women of the same chronological age as compared to premenopausal women. And this might be important, for example, for identifying the transition into menopause, because that's a time where some treatments could be given to delay the onset that has been shown to have health benefits. So in the final few minutes, I want to tell you about more recent efforts that we have on trying to take all of this data and try to work towards eventually integrating it all together to hopefully get a more holistic view of human health. And so far I showed you various projects in which we focused on one particular question, addressing typically one particular data or developing models for a particular task. And in recent years, we're seeing kind of a change in or new types of approaches being developed in the field of AI, whereby you can take just the data itself and without labels work on what is called a foundation model. It's called a foundation model because it's not developed for any particular task. It's developed to kind of learn the statistics and the patterns in the data, and then you could fine-tune it and use it for specific downstream tasks. For example, a large language model, LLM, is just trained on a big corpus of textual data. It learns a model of language, and then one application of that could be a chatbot like ChatGPT. So the idea is to try and take all of our data and try to integrate them together into a single model that would have many different applications. And just to give you an idea of how this works, so rather than, as in the past, taking label data, like for example, taking digit data and saying that the first two digits are 0, the next one is 7, the next one is 2, and having those labels and trying to predict those, what the new type of approaches in AI try to do is a framework called contrastive learning, whereby you would take, for example, the two top images, and without saying that these are 0, you just say that these are similar to each other. These should be the same. And the two digits at the bottom, 7 and 2, are different, so they should be different. So what you ask the model to do is you run a model, and you tell it that you want the model to embed the images at the top in a space such that they would be close to each other, and the two other images to be far from each other. There's a mathematical framework to that. There's other approaches in which you can do that. For example, you can take a picture of a dog or a picture of a chair. You can break it down into, you can take different slices of the dog. It's still a dog, and you tell the model that these are similar to each other. Similarly, different patches of the chair are similar to each other, but the chair and the dog should be different. So by doing that, and doing it also for text and images, and what's called DALL-E that you probably play with in OpenAI, what you do is you take the legend, and you take the caption, you take the image, and basically everything on the diagonal are what's called positive examples. The text corresponds to the image, and everything on the off diagonal is should be different, because it's an image and a caption that do not correspond to each other. And just by doing that, you're basically aligning the text to an image, such that then you could just give a text and you would get back a generated image that pretty much corresponds quite well to the image. So the idea that we're trying to follow is kind of the same, to try and take all of these different data modalities that we have on a person. They each are related to each other in the sense that they each inform us of a different aspect of human health, but they might be measuring different body systems, and still we want to put them together. So the one example that I'll give is a recent work that we did on looking at the cardiovascular system, and looking at two different data modalities that we have. The retina scans on the one hand that I showed you before, that shows all the intricacies of the blood vessels, and the only non-invasive way in which you can see blood vessels in the body. And on the right-hand side, an ultrasound of the carotids, also informing us of cardiovascular health, but they do so in two very different ways. The measurements are done from different parts of the body, obviously, and also by different methodologies, different technologies. So the idea here is to build a model where the only thing that we do using this contrastive learning framework is that when we have a retina scan and a carotid ultrasound that come from the same person, we tell the model these should be embedded to be close to each other. And then we take retina scans and carotid ultrasound of different people, and we tell the model these actually should be distinct from each other. We can also do that not just on the same images, the images from the same person at the same time point, but we can also do it across different time points. So we can take the retina scan of a person done at baseline, and the retina scan of the same person profiled 2 or 4 years later, and tell the model these should be similar to each other, whereas different images from different people should be different from each other. And when we do that, similar to kind of the magic that happens with these models in DALL-E with text and images and so on, we find that we can do tasks that medical doctors or humans probably can't do, which is to take just the retina scan of a person and then have it rank all the carotid ultrasounds, all the 10,000 images that we have, And we see that the accuracy is not perfect, but if you look at the blue curve, for example, here at the top, then within the top 100 images out of the 10,000 images, in 70% of the cases, it will put the corresponding ultrasound carotid image of the person within the top 100 images. So this is something that probably you can't see, and what the model is doing is probably picking up on some particular patterns in that person and somehow aligning those. So the idea is that these types of representations could be useful for many downstream tasks. We show that it helps us to predict future cardiovascular events, and also to predict responders and non-responders in clinical trials. So with that, I'll just kind of go back to the high level, which is after seeing what I think is a great utility in being able to learn many things from these types of population level cohorts. We're trying to now significantly expand this cohort on multiple different dimensions, growing the number of people to hopefully reach 100,000, looking at more geographies, more countries, to look at more cultural and ethnic and genetic diversity. Also profile more phenotypes that we are not profiling today, and looking at more diseases. And I believe that these types of models also, when applied to these types of next generation AI models, will really take us to the next level in terms of precision medicine. And with that, I'll just put the acknowledgement slide of all the people in my group who contributed and led this research. And thanks for your attention. Happy to take any questions. Thank you.
Thank you, Eran. A very nice and inspireful talk. So let's open the session for questions from the public. There's a question over here.
Hi, thank you for the talk. Monika Lewinska from Gubra. I have a quick question about the cause and consequence of your bacteria SNP and BMI. Where is the egg and where is the chicken? Is that the dietary choices that led to high BMI that then preferentially caused the growth of those SNP harboring bacteria, or other way around, and whether you did some antibiotic therapies to prove that, or fecal matter transfer or something?
Yeah, so that's a really great question, and that's the million-dollar question in this field, the cause and the effect. Unlike in genetics, when we do these associations in the microbiome, because it's an environmental factor, maybe it's actually affected by BMI. So ultimately, the way to show that is with experiments, which we are in the process of doing. This will take time, but we're now in the process of isolating the relevant bacteria from people who harbor the strains and the SNPs that we think help with weight loss. After isolating these bacteria, we want to give them in clinical trials as probiotics to see if they indeed induce weight loss. And this is ultimately what we have to do and are working to do. I think that all the evidence points to the fact that a lot of these actually probably would be causal, which is why we're putting the effort in doing these experiments. And the reason I think that is because I think it's otherwise very hard to imagine how a trait like a BMI or other traits would actually cause the bacteria to evolve genetically such that there would be concordant changes in multiple people at the genetic level. These are things that we only see, and actually we do see, when you give antibiotics, and we know that that happens. That's a very strong selective pressure, but I don't believe that there is such a selective pressure in a trait like BMI. And the mechanism I think that's going on is more that you kind of randomly get your bacteria from the environment. Some bacteria have a propensity for obesity, and some people, got a few of those unlucky ones, but then they got a few other ones that may be good, and it all kind of balances out to give an overall propensity for weight loss or weight gain by the microbiome. Very similar to how we think about genetics, that you kind of randomly got your different variants, and some increase and some decrease your propensity for some trait like obesity, and overall there is some net effect. I think that's the simpler explanation, but that's a long answer to say in the end that we have to prove that, and we're working on proving it.
And maybe just to follow up, have you considered including in that modeling whether the patients or subjects live together in the same household, therefore exposed to same or similar dietary, dietary causes, I don't know, preservatives, dyes, whatnot, that could be environmental or coming from the same household?
Yeah, so we do look at we try to look at other confounding variables that we have, and sometimes we have people living in the same household, sometimes not, but we try to collect all of these factors, and the SNPs that I showed are the SNPs that we found beyond the confounding factors, so we can't explain them by anything else that we measured.
There is another question here in the first row.
Tunepes from the University of Copenhagen. I was interested or curious about the foundation models. Are you building them based on larger models that you are fine-tuning, or is it do you have enough data to just build them from scratch to sort of get what you are sort of aiming at?
Good question. So typically with the we typically try to take pre-trained models as a starting point, and we find that often actually helps. So for example, with the imaging data, we use Dino2 as a starting point of a pre-trained model, and then we fine-tune it on the images that we have. With other types of data, like the continuous glucose monitor or the microbiome, where we're also trying to build these models, our data is the largest, so there's no other data. We're just using that. And it's working. I can't tell you if we had 10 times or 100 times more data, whether that would get us to the next big leap. Probably it would help. So yeah, whenever possible, we're trying to take pre-trained models from the literature.
There's another question over here.
Hi, very interesting talk. Just building on this topic of AI models in this context, and of course in personalized medicine, I'm kind of not worried, but a little bit iffy when it comes to these models, because at the end of the day, it's what you're putting into it, what you're asking it to do, that's the output that you're getting. So are we asking the right questions? Are we training it the way that we want it to get the answers that we're getting? And is it getting a little bit out of hand in terms of we're feeding data to it, but there's going to be a point where we don't know what we're getting out of it, and maybe statistics with it, the relevance of it, what scientific questions we also want to ask. The model is not really thinking for us, right? So of course it's a valuable tool, but do we have to be careful moving forward in what is it actually telling us? What do you think about this?
Yeah, I think it's a very fair question. So, I think prior to these, or the models that are not these foundation models are typically developed for one particular task, and then what you have a hypothesis, you're asking a specific question. I think with these foundation models, initially, you're trying to build a model that just captures the patterns in the data. But then you're right that, you don't stop there, but then you go and you ask, okay, so what is this good for? So these are the types of things that we're trying to work on, to show that in various downstream tasks, then you can use these models. So I showed a little bit that the model I showed for the carotid ultrasound and the retina scans could help us and predict better future cardiovascular events. So that would be something it's relevant for. But ultimately, like with the large language models, you want to show that they don't just learn a model of language, but then you can do something useful with it. And in language models, we know that you can do many useful things with it. I think here we're still in early days. We're starting to see some signs, but I think this is where the field will be working on to try and show what applications these models are good for.
Great, thanks.
So there is another question here in the first row. Another question here. And we'll take this as the last question. Here.
Thank you very much. Absolutely fascinating. So your organ-specific clocks, I just wonder in terms of, people don't care about, individual, my kidney is older or my brain is older. So ultimately, do you have an insight into what is the kind of clinical or longevity or healthy longevity relevance of these different systems for your overall kind of health and survival? I mean, it's
Yeah, so I hinted a little bit to that typically when we find that these clocks are disrupted in people, meaning that you're older compared to your peers of the same chronological age in one particular system, we show that that has clinical relevance in the sense that then those people are at higher risk for disease, but not just any disease. Typically, it would be a disease related to the body system on which the clock was developed. So in that sense, the clock for the liver is telling us a little bit about the risk for developing liver disease, fatty liver disease, and so on. So that's where we see the relevance.
Yeah, sorry if I wasn't clear. I meant more the relative importance of each of the organ systems for your overall survival. So
Yeah, overall survival. We don't have data on overall survival for this population, but I would say that I think any of these diseases that we're finding you have elevated risk for, then first of all, you are at higher risk from suffering from that disease, and probably many of these diseases also, that's known in the literature, leads to a reduction in overall survival.
Perfect. Thanks, Eran. One more
Thank you.
time for a fantastic talk. Let's give an applause to him.
Thank you.
Thanks.